Okay, and with that, I would just want to present to you the, Matt Seidensticker of the Montana Moth Project, and I had the other part of that written down somewhere. You say it. The organization.
Northern Rockies Research and Educational Services is my little nonprofit.
That was too much for my brain. Thank you, Matt. Do I give this to him?
Okay, can everybody hear me now? Yeah, is that good? Yep. All right. Well, thank you, everyone. It's a pleasure to be here. It's always a fun night when I get to talk about moths. And so hopefully we'll make some connections tonight between moths and birds. Of course, we all know that birds eat a lot of moths. So what I want to do here tonight is I just want to give you just some brief background information on what is a moth, some natural history, things like that. And then we'll talk about how to moth. So there is such a thing as mothing and moth watching, and in fact there's a Facebook page that's dedicated to all this. So not unlike bird watching or birding, and kind of tell you how do we find moths? How do you do that? And then I'll weave in sort of some of our research that we've been doing with moths, and then I'll kind of end by talking about the Montana Moth Project, which is the flagship program of my little nonprofit. And so this is about moths according to me. And so just general natural history. So when we're talking about butterflies and moths, we're talking about the order Lepidoptera, right? Leps, as people call it. Now, I had kind of this circuitous route in my career. I used to work with owls and Denver Holt and the Owl Research Institute, and I kind of gravitated into moths. But when I was working with the owls, we would always say that owls are probably one of the most recognizable animals on the planet, right? Like everybody kind of knows what an owl is, right? Well, I feel like it's the same way with leps, so to speak. So certainly everybody knows what a butterfly is, for example, right? And kind of by default, you know what a moth is. So leps, moths, and butterflies are, they're among the most easily recognizable insects that we have out there. And, they're a single lineage, right? So they're related, they're cousins. And there's a bunch of different characteristics that sort of define them. But the big ones are, and let's see if I can do this without, are the scales, right? So they have these modified hairs, scale-like hairs on their wings, right? Which is what gives them their colors and their patterns and things like that. And then they used to call it a proboscis. So all butterflies and moths have it's sort of think of it as kind of like a drinking straw that they curl up between their palps, right? And they've now changed it to call it a hostelum, and we won't go into the semantics of all that, but a hostelum and a proboscis are the same thing. So those are the two things that really define moths and butterflies from other insects. Does anybody know the easiest way to tell a butterfly from a moth? That's right. Well, that's a good one.
Feathered antennas. Feathered antennas.
Yes, it has to do with their antennas. Yes, right. Okay, so you're getting close. Yeah. So, right. So with butterflies, they'll always have this little club-like structure on the end of their antenna. At least everything north of Mexico will. There's some tropical moths that don't have that. Whereas moths will have this, as you said, either a feathery antenna or just kind of a filiform antenna, right? They'll never have that little club-like structure on the antenna. And so you can see this a lot of times, even in the field with your binoculars or whatever, and there are day-flying moths. And so if you see something flitting around, you kind of zoom in on it. If it's got the clubs, it's a butterfly. If it doesn't, it's a moth. Now, an even better way, and what we use sometimes if we have specimens, and sometimes if you have a live specimen, you can see this, and this is something that differentiates moths from butterflies, is this frenulum. And so moths have a linking bristle between their forewing and their hind wing that kind of links it to act as like a single airfoil, whereas butterflies don't have that, right? So if you were able to flip the moth over and look at these linking bristles, you could definitively say, oh, that's a moth, it's not a butterfly. And we can also tell sexes using this sometimes too. We can use the antenna to tell sexes. A lot of times the male will have the really bipectinate feathery antenna and the females less so. We can also use that with the frenulum, where the males will just have one linking bristle and the females will have more than one. Now, there was this big debate for a lot of years between the moth people and the butterfly people, where the butterfly people are like, oh no, you know, moths are just nighttime flying butterflies, and the moth people would say, oh no, butterflies are, you know, daytime flying moths. And there was this big, like, you know, back and forth going on for years and years, right? Well, Kawahara, and it's hard to see here, Alito Kawahara at the McGuire Center, he did this massive phylogenetic study with, like, I think it was almost 30,000 specimens from, I believe it was right around 40 different, like, collections throughout the world. And he did this massive phylogenetic study. And bottom line so the oldest Lepidoptera fossil we know of is about 220 million years old. So way back in, like, even the Triassic, kind of almost Jurassic era. And during this phylogenomic study, he concluded that it was most likely that this was some sort of moth type of insect, right, versus a butterfly. And then he also determined that butterflies split off from these ancient lineages of moths about 110 million years ago, which coincided with the proliferation of flowering plants. And so basically, the debate settled. Butterflies are just day-flying moths that filled that diurnal niche when all the flowering plants radiated. And so he kind of settled that for us. And what's interesting enough was he also determined that split happened somewhere either in the western United States or Central America is where that split of butterflies from moths happened 110 million years ago. So just a little tidbit there. We won't get too deep into that. Now, Lepidoptera is one of the most speciose. You can't see that, but they're the most species-rich orders in the world. So currently, they're being discovered all the time. Let's say there's 1.1 million described insects right now, approximately. Most people think, okay, the beetles, right, Coleoptera, are kind of the most species-rich. There's maybe 400, 500,000 described species right now, and then it's sort of a debate whether it's the Hymenoptera, the bees and wasps and things, or the flies that are kind of come in second. I think eventually we're going to find that the bees and wasps are probably the most speciose, and then in third place it's solidly the Lepidoptera. And so worldwide right now, there's probably there we go. There's approximately 160,000 described Lepidoptera worldwide at this point. The interesting thing about this now is that 90% of those are moths. So currently so moths outnumber butterflies diversity-wise. Some people say 9 to 1, some people say 15 to 1. It's hard to know. Most of the experts think that this is probably more like 300,000. Just moths that are described, because there's, moths sitting in collections that haven't been described yet and things, and we'll talk about that a little bit later. But very, very diverse group. So we're talking maybe 140, 150,000 described moths currently, and then approximately 20 to 30,000 butterflies worldwide. So a lot more moths out there in the world than there are butterflies. Now they have this, what we call complete metamorphosis, right? So they go from the egg, and this is based off a monarch, not a moth, but it's the same thing. And then to the young larva, which we call the caterpillars, right? And then these caterpillars go through different instars, and then they eventually reach their last instar here, which is that classic thing. And then they'll go into the pupa, which in butterflies we call a chrysalis, and moths we call it a cocoon generally. And then they emerge as adults, and so they have this complete metamorphosis life cycle. Now, most Lepidoptera, almost all Lepidoptera, there's a few that will feed on blood, some drink tears, but by and large, 99.9% of them feed on plants, right? And in fact, they're the most species-rich lineage, and we talked about how they diversified with the radiation of flowering plants and things like that. They're the most species-rich lineage to depend on flowering plants, and they're only rivaled by a few of these beetle clades. And we'll get into this a lot more as we're talking about why moths matter. And so the adults will also feed, right? So a lot of moths, there's some, like if you get a chance to look at some of the specimens I brought, the one in the cardboard box, the really big polyphemus moth, they don't feed as adults. So they go through their life cycle, they breed, and they basically die. They don't even have, like, they just have like a very, like nubbins of a hostel, and they don't even feed. But most of them do. Even the little dinky tiny ones, they'll feed, and they'll sip nectar and water, things like that, dew from plants. But they don't grow. Once they eclose, that's the size they are. And we talked about some don't feed at all. And then the females call with pheromones, right? So these volatile chemicals that the female emits, the males come in, find the females, and they breed, and that's how they reproduce. Now, many females are actually wingless. So there's certain species where the females don't even have wings. They just let out the pheromones, the males come in and reproduce, and they're just sort of like stuck to their host plant. Now, this is going to be interesting when we start talking about why moths matter too. So according to Powell and Ockler, who were two giants in the field, they claim that the average moth lays about 200 to 600 eggs per female. Okay, some do thousands, right? And we'll talk about this more. Keep this in mind when we start talking about why moths matter a little bit more. And then like with a lot of things, we differentiate species based off their genitalia. And I'll show you a quick slide here of especially these smaller little guys. So if you look at those really tiny little, we call them micro moths that we have over there, they're really hard to identify sometimes without looking at their genitalia. And there's other groups of like this little geometer moth, the Eupathecia. And so this is what a genitalia looks like. So this is a male genitalia. Here's the phallus, and here's the valves and things like this. And so this is found on the very end of the abdomen here. And we'll talk about this a little bit more too, but that's how we describe species. If you want like a solid identification, that's how it's done. Now, like with all animals, they have some really cool adaptations, right? And so for years we knew this evolutionary back and forth, if you will, between moths and bats, right? They both occur at night. Bats are eating a lot of moths. Well, moths kind of come up with these cool adaptations that they can hear bats, right? So a lot of moths actually have tympana. They have simple ears on their abdomen a lot of times, or their thorax here, that they can actually detect bat ultrasound or echolocation calls, which is really, really neat. And so we knew that a lot of these moths could hear things. Well, the Smithsonian, there's this one moth in particular, and this was really common in like the tiger moths and certain groups. This Bertholdia trigona is in the tiger moth family. And they figured this was doing something different versus not just hearing it, but they suspected that it was doing something else to counter this bat echolocation. And so what they did was they took a bunch of these moths, they stuck them in a dark room, hung them from basically like fishing line or whatever, and they pierced the tympana of some and they left others intact, and they released a bunch of bats in the room. And then they had all these recording devices going as well. And basically what happened was all the moths that had their tympana pierced were being depredated, and the ones that weren't. And during their recording device, what they found was that these moths were emitting their own, so they'd hear the bat echolocation and they'd start emitting their own ultrasonic clicking sounds, thousands per second. And basically determined it's a sonar jamming thing. So they're not only actively hearing them, but they're actively like jamming the bats' echolocation as well, which is pretty cool. And so and this is becoming known more and more in a lot of tiger moths and other moths as well, that it's not just we hear them and we take evasive maneuvers, but we're actually like actively like jamming their location or echolocation, which is pretty cool. So, and this is a picture of one. So we actually have this. We found this moth. This is mainly a moth that's known from like the Southwest, but we find this moth in the extreme southeast corner of Montana, out near Ekalaka and that area. This is a picture of one of the specimens that we collected out there, and so this whole evolutionary arms race is playing out right here in our home state of Montana, which is pretty cool. And so they also have, some moths have predator mimicry, right? So there's this really tiny moth, a group of moths, the metalmark moths, right? So these things are really super tiny. Well, what they do is they mimic the courtship dances of jumping spiders. And so this is one of these metalmark moths where they pronate their wings up. So here's the forewing, here's the hind wing. And so when they feel startled by a predator, they do that to mimic a jumping spider. And in fact, they're so good at it, there's this app we'll talk about called Leps by Field Guide. It's an AI app that you can use. Somebody had put this metalmark moth in, and the AI thought it was a jumping spider. So they use, a lot of the moths' maculations and patterns are meant for camouflaging and things like that. In this case, it's a mimicry to avoid predation. And so then another cool thing, this bogong moth in Australia. So this is a moth that migrates. So it breeds down in the lowlands of Victoria here, okay, during the winter. And then in the spring, it goes up here. It migrates up into these, I guess they call them the Australian Alps, or in the southern Queensland. It's about a 1,200 kilometer journey that they make to go up into these. And this is they go into these caves and they estivate, which is basically like summer torpor. Think of it that way, versus like winter hibernation. And if this is a picture, these are all moths right here that are in this cave. And then once they estivate in the summer, they come back down in the fall and they breed. Well, for years they were like, how are they doing this? Like, how do they know, how do they navigate up there, right? Well, they did an experiment where they basically have these wooden boxes, and they were catching these moths on their way up, so in these areas here. They have these wooden boxes with magnetic coils. They caught some moths, they put them in the boxes, they flipped the magnetic coil on, and at first the moths were just kind of like spinning around. And so then they put a silhouette of the mountain range at one end of the box, and they flipped it on. And as soon as they did that, the moths oriented themselves to face that silhouette of the mountains. And so really what they showed in this experiment was it was one of the first, like, we always suspected with like monarchs and like the painted ladies that fly over the Mediterranean and stuff, that they're using some sort of magnetic sense. And we know that in birds, right? Birds use, magnetic fields and stuff to migrate. This was the first time that they really sort of had good evidence for that in an insect. And as a follow-up study, we talked about how we still don't know why our moths attracted to light, or are they disoriented by light? Like, why do they come to light, right? So they did a follow-up study here where they took some moths and they exposed them to the natural sky and sunlight, and then they took them into planetariums and did the same thing. And then they used the magnetic thing. And basically, long story short, is they found that yes, they're using this magnetic field to orient themselves directionally, but they're also using these visual cues that they suspect are these, starlight, moonlight, things like that, to orient. And every time that they would try to, change up the sky or whatever in the planetarium, the moths would always orient themselves to what the constellation would be, or the night sky would be heading north or heading south and things. So Really interesting thing. I suspect, like with the Army cutworms that we have, we'll talk about a little bit here in North America, are probably doing similar things. And so we'll speed up through this a little bit. We're almost done with this natural history part. Okay, so why do moths matter? Why do we care about moths? The first thing is we talked about they're inextricably linked to plants throughout their whole life cycle. And Dave Wagner, who's a famous lepidopterist, he'll tell you that they're the predominant herbivores out there. And we'll go back to that. Think of that 200 to 600 eggs per female thing, right? Powell and Opler also stated that it only takes 2 eggs per female to reach adulthood and reproduce themselves to maintain stable populations. And so if you follow that logic and extrapolate that, there must be, I don't know, billions, trillions, a lot of caterpillars and things out on this landscape, right? And so Wagner would say that whether you measure it by diversity, abundance, or biomass, Lepidoptera are probably the predominant herbivore on the landscape in just about any ecosystem. Now, some of them, right? Some of them, they're kind of pests, right? So some of them we know they'll feed on, they'll cause damage to crops and things like that. Like this Army cutworm moth here that we'll talk about more. This Douglas fir tussock moth, which caused a bunch of damage to some fir trees north of Missoula here a few years ago. But most of them, 99%, there's a handful of pest species, but 99% probably of all Lepidoptera rely on native plants to complete their life cycles and things. And so, very important as herbivores on the landscape. The adults are also pollinators, and for years they were really underappreciated and overlooked pollinators. And part of that is just how do you study something that's out at night, right? We knew that some of the larger sphingid moths and what they call the hummingbird hawk moths, and I've got a white line sphinx over there. They're sort of crepuscular. You can see them in your gardens and stuff doing their thing. But we didn't know really the extent of how much these moths were pollinating. Here's a shot that Christy Du Bois took of this is actually a biocontrol moth, but it's the leafy spurge hawk moth. You can see it in there. Well, a few years ago, let's see here. Oops. So a few years ago, some folks did these meta studies where they looked at all, they started to get interested in this. Okay, like we suspected moths, a lot more moths are out there doing their pollination than we think, and so they did these meta studies where they brought together all these literature reviews and stuff. And, like McGregor found that there were 75 plant families of 289 species of plants from 21 moth families that were being pollinated. Hahn and Burrell found similar things, and what they were starting to see just based off of what was reported is that there was a lot of complementarity, if you will, or sort of redundancy happening, where they were finding that the moths were visiting a lot of the same flowers as the bees and vice versa, and then there was some specialization involved. Well, what really changed the game for us was this little thing called, and a lot of these studies, so moths, they're not collecting the pollen, right, like bees. They're incidental pollinators, we call them. And so some of these studies were finding that they were promoting better gene flow between plant populations. So you can imagine like a ground nesting bee, a lot of times it'll only forage within several hundred meters of where it's nesting, right? Whereas moths are very mobile, they can travel, miles, kilometers, or whatever, and so they're promoting genetic diversity between plant populations based off of this long distance plant pollen dispersal, right? And that they may even be more efficient pollinators because they're not taking as much pollen as they're maybe leaving behind, right? So the bees are stuffing their things, and it's just maybe more efficient with moths. And so it really changed the game, and don't get overwhelmed with this thing called DNA barcoding. And so barcoding is there's gene regions in animals and plants that they're short DNA sequences that are so conserved within a species but variable across species that we can literally like take a piece of tissue or a pollen grain in this example and amplify that gene region and tell you and compare it to like a known sequence, and in a lot of cases determine what plant that pollen grain came from, right? So this really changed the game for us in terms of moth pollination, because then we could collect moths, we could swab their bodies, we could take their proboscis, and we could amplify that DNA and tell what they were visiting. And so this McGregor, he did this sort of validation study where he compared what we used to do, which was light microscopy. So you take pollen and you'd expose it to different UV lights, and you'd look at the morphology and this and that, and you'd identify it, which took like lots of expertise, tons of man hours. Whereas with this DNA barcoding, you just collect it, send it to the lab, they amplify it, you compare it. And so he did this comparison between the two, and what he found was that even though they were comparable, the DNA barcoding was better at like detecting the rarer pollen and rarer plant interactions at a much lower cost, right? So this really opened up the thing. So we could go out at night, and we've done some of this on MPG Ranch. So here's the hostelum, right? This is all pollen on this moth's hostelum. This is all a pollen load here on its body. And so we did a little study here that we're just getting ready to submit the paper this month. We looked at some of the lower elevation restoration habitats on MPG Ranch where they had done plantings and things like that, and we collected bees and moths. And we did this barcoding to compare them and to build these networks, these pollination networks. And here's just an example of some of the networks that we found, right? So here's the diurnal is the yellow, and then the nocturnal is here. And as you can see, here's the interactions, right? So here's all the diurnal interaction. These are all the plants and stuff. And we won't get too bogged down in this, but basically what we found is that all of these, the bees and the moths were visiting 53 plant species from 23 families. And most of the interactions were coming from the Asteraceae or whatever, like the sunflowers, things like that. But what's cool here is that moths comprise 60% of all the interactions in these networks, right? And even visiting more plants than the diurnal pollinators. And so for years, moths were pigeonholed into this what we call pollination syndrome, where it was like, oh, moths are only pollinating white flowers that are heavily scented that open at night, right? Well, what we found here is that, and there's a ton of other studies that are coming out, is that, and we showed you that little graph with all the connections, like the moths are visiting a lot more flowers than what the syndrome would predict. And they're visiting a lot of the same flowers as the bees are, and kind of vice versa. And even there's some specialization where the moths are visiting like the longer corolla flowers that the bees maybe not necessarily doing. So to sum it all up, basically what all this means is that there's a 24-hour pollination service happening out of the landscape, right? And for years and years and years, we were just looking at like the bees. And this has direct application, practical application in terms of like our restoration mixes that we're planting. So a lot of the restoration mixes that we replant were geared towards bees, right? So they were all focused on the diurnal side of things. This is showing that basically we might need to rethink how we're like planting our restored habitats to take into account this other side of the pollination thing. And so that's a long way of saying that moths are out there doing a lot of pollination, and they're providing a lot of resiliency to these pollination systems. Here's a cool alluvial graph that just kind of shows that some of these things you know, like, of course, these are all like, most of these are like introduced, like that's leafy spurs. But they started out like, you know, with diurnal, and then they'd be visited more than nocturnal, and they'd be visited by both. But this has really opened up a ton of stuff for us, and there's a bunch of studies coming out. Oh, and we also did this with a rare plant in the Pryor Mountains, and we found that, oh geez, I'm really skipping around, that this is Physaria pachyphylla. It's a threatened bladder pod in the Pryor Mountains, and we found its pollen. We didn't really know what was pollinating it, and we still have a lot to work to do, but we found its pollen on these 4 moths. So these moths are you know, interacting with rare plants even. And so this is just to give you an example of like a lot of the studies that have come out, right? Like they do a large range of crops even, systematically overlooked, excluded components, right? This study found that moths actually contributed more to fruit set in apple orchards than diurnal bees and things like that. And so this is just, the point here that I want to get across to you is that more and more stuff is coming out, but it's a 24-hour thing that's happening out in the landscape, and moths play a huge role in that. And so as we move forward in talking about conservation of pollinators and restoration of habitats, we can no longer overlook what the moth roles are in that. Highly important. And of course, they're important food resources, right? So, let's see here. Oh yeah. So 16 of the 20 major bird families eat caterpillars to some extent, and mainly to provision their young. And even things all the way up into grizzly bears. So we talk about, Steve knows about this in Glacier Park, and there's some in the Mission Mountains too, where the Army cutworm moths they do it like the bogong moth. They go up into these high elevation scree fields in the summer and they estivate. And the grizzly bears go up into these scree fields and they turn over rocks and they just eat gobs of these things. And maybe you can correct me if I'm wrong, but there was a master's student, right, that did some work a few years ago in Glacier, and I can't remember the number of moths it is, but basically like there's an X number of moths that's like basically eating like a double cheeseburger for the grizzly bear, right? So very high caloric value for these. And to go back to Wagner, right? So again, Wagner would tell you that whether you measure it by diversity, biomass, or abundance, moths transfer probably more energy from plants, and this is just plant-eating insects, more energy from plants into higher trophic levels than any other plant-eating insects. So hugely important food resources. And we know bats eat a lot of moths, along with a lot of other things. And so basically what you do is you take this, let's see here, oh, this is just a picture of a poor whale that we think was, that might be a moth in its mouth. It just threw it in there for, we'll talk about MPG Ranch a lot more here in a second. So you add this all up, right? So we've got, why do moths matter, right? They have this inextricable link to plants. They're predominant herbivores in the landscape. They're important pollinators, key food resources. And then you add that into this staggering diversity, right? We already talked about, like, you know, they're outnumbering bees like 5 to 1, butterflies 10 to 1, a lot of different things. There's so many moths out there with pretty well-studied taxonomy. So for by and large, most moths that we catch, we can kind of look at them and say, oh yeah, that's what that probably is, right? Or somebody knows what that is. Along with this relative ease of sampling, we'll talk about this more. Basically, it all adds up to that they're suburb ecological indicators, right? They're very, very important. So we can, you know, if you have an area that's got a diversity of plants and native plants and things like that, you're probably going to have a real diversity of moths, which in turn you're going to have a diversity of birds and other animals and things like that. And so they can really serve as a barometer of environmental change. And what's really cool is this relative ease of sampling, which we'll talk about a little bit more. It doesn't take much to sample moths. And so, you know, like a lot of things, moths are declining, right? And we're kind of worried about them. Insect declines are a tricky thing. I'm sure we've all heard about the, you know, insect apocalypse and the huge study out of Germany that kind of started all that. It's a little bit touchy, you know. I mean, traditionally, entomologists didn't count stuff. We were worried about pest management and describing new species, and so a lot of areas were lacking the data. There are some areas like the UK that have a 50-year moth study going on, and they've clearly showed declines in the moth populations there. Like this garden tiger has declined like 70% in the UK. It used to be really abundant. There's evidence of hawk moth, declines in the Northeast. And is there a general decline worldwide? Well, because of this lack of data, we rely on these meta-analyses and literature reviews and things. And certainly, you know, a lot of these articles and more studies that are bringing together some of the longer-term studies are definitely showing a trend towards declines in a lot of things. And butterflies or moths are no exception to that. Now, why is that? It's your usual host of characters, right? Habitat destruction, chemical pollution. A biggie for moths that we're starting to find out is this light pollution, right? That's been a biggie. There's been studies that have shown that it, you know, kind of alters their reproductive cycles and things like that. Climate change, I think, is going to be obviously a huge one for all insects because they're prone to desiccation and things like that. And then your consequences are kind of the same thing, right? You might lose plant communities, disruption of food webs, biological deserts. We have a long-term study going on at MPG Ranch that's looking at local climatic factors related to this. And we're only in our 7th year, so we can't really make any inferences yet. But there are folks out there trying to gather long-term data sets. But as you can imagine, insects are hard. You know, there's a lot of them, and counting them is hard. It's just hard. But I think anybody that, I mean, we're all out in the woods and the landscape a lot, right? And you hear about the windshield phenomenon, right? That maybe there's not as many bugs spotted on your windshield as when you were growing up. And, I mean, I think I was just having this conversation with my, and I don't want to get on a tangent with my excavator, and I was like, you know, because we had like 3 inches of rain in Missoula in December, right? And it's like, what? And I said, you know, I don't know, we can debate the whole climate change thing and whether you believe it and why, was it caused and this and that, but nobody can deny, right, what they see with their own eyes. And things are changing, right? And I think, you know, if you notice that hey, maybe there's not enough as many insects on my windshield, it's probably true. Whether we have the hard data to support it, probably not. Anyway, so now we're going to shift gears a little bit, and I'm going to speed it up here so I don't hold you too long. So moths, there's a seasonality to moths, right? So there's moths that fly in the spring, there's moths that fly in the summer, and there's moths that fly in the fall, right? And so these guys here are spring moths, and you can see them flying right now. So I think this thing's on a timer, actually. I might have screwed something up here. So these Agyra, it's a genus Agyra. They're flying right now. They're spring moths. I think I got it on a timer somehow. And then Pleuramaloida, Lasiocampini, these are Orthosia. And so there's this whole, the point of this is there's just this whole set of species that fly in the spring. Okay, and so these are all flying as we speak right now. And this is going to link into birding here in a second. This is a Lithophany, and you'll notice whose pictures are Christy's pictures and my pictures. Mine are terrible. Yours are great. This is another one that's flying right now. This whole genus called Lithophany. They overwinter as adults, and then they come out in the spring and breed. And then they'll fly again in the fall. the next generation. Yeah, and then Feralia, here's another lithophony. And so these are all just spring flying moths. So we're just going through the seasonality of moths. So these are all things you can see right now, flying around. This is my favorite. There's no common name, but it's called Varencia conchiformis. I call it the galaxy moth because it's got this kind of like nebula, these cool things. These, tiger moths, stretchia, these are all flying, right? So you have this whole set of moths flying, and I think it might work better when you switch it. Yeah. Pero is another one. Zales are another kind of a spring moth. And then we have, so starting in about early May to mid-May, you'll start seeing this guy. This is a Hyalophora silk moth. It's interesting, we are in kind of a hybridization zone between Wolveri and Euryalus, and so this looks like a true Euryalus, but you'll see these guys a lot, just hanging out on old barns and things like that. And they're females can be the size of like a salad plate, pretty large. Males are a little bit smaller, but you'll see these coming out in May. Sphinx moths start coming out in late spring, so this is a one-eyed sphinx. I have one of these over here. This is called a, I don't know if there's a common name for this one, actually. Smerinthus. Sphinx moths, Sphinx vashti. And there's your white line sphinx. So this is one you'll see in your gardens a lot, crepuscularly, flying around. And then sheep moths, this is a diurnal moth, and you'll see this starting in May, up in like alpine or montane meadows, things like that. And so these are all spring moths, right? And so there's a seasonality to it. Go ahead. And then we get into our summer moths. So then, starting in like towards the solstice, middle of June, we start seeing this whole different set now of these, what we call Apamia. These are all a genus called Apamia. And there's a lot of different species here. This guy here, Inordinata, this is one that when I talked about the MPG work, this is a huge pollinator. This was involved in a lot of different interactions. And so we start seeing our Drasteria moths, which are tiger moths, another Drasteria, right? And so these are all our summer moths, Zotheca and stuff. Keep going. These are diurnal moths, these forester moths, and you'll see Glacier Park's got a lot of good place to find these forester moths. And then these things we call Plusii moths, or the looper moths. Somebody showed me a looper moth earlier that they brought in. The Cynipolia, more tiger moths. And so as we get into the summer months, that's when things really start ramping up. And then somebody was mentioning cutworm moths, right? And so cutworm moths, it's a really broad category, and this is a genus called Euzoa, and there's several hundred species in this genus, and they're all considered cutworms. And so they're larvae, they're in the soil, and they come up and they feed on the bases of grasses, or some climb up and feed on the buds and things like that. But a huge genus, and so the summer is really when we start seeing our little micro moths, right? Our tortricid moths, and you can just kind of roll through these. More of these. Here's a crambid moth, and so the summer is when we see the greatest abundance and diversity, right? Because these are ectotherms. They kind of time these are plume moths. We suspect these are really big pollinators. And so the summer, Abagrotis, Parabagrotis, Zestia, Dargida, and you just scroll through these and we'll get through then we see the autumn moths. Okay, so this is a Lambdina, a whole new set, right? So you can just scroll through these. So the whole point of this is there's a seasonality. Ghost moths, which are really cool. Spring, summer. Catocala is a really cool one that comes out in August, you can see. And we'll just get to these graphs and I'll just show you a visual to kind of get this point across. So this is just a couple of years of data from our moth monitoring at MPG Ranch. And so you can kind of see where, this summertime, right, is when we see our greatest abundance. and diversity is in the summer. But you can also maybe see a few little peaks, you know, like here in the spring moths or whatever. And then there's the fall flying moths. You can almost see this. And the next slide might even show this a little bit better. So all the blue here, and if we had the months here, you know, this would be like April through like mid-June. You know, these are all the set of genre of spring flying moths, and then you have your summer moths, and you have a distinct kind of change in your fall flying moths. So, point is, there's a seasonality to moths, and we'll talk about this as we start making the link now between bird watching and moth watching, right? So I'm sure you're all dying, like, how can I go out and do some mothing, right? You know, you're probably like, oh, when's this guy gonna stop talking? So I pull this on the younger kids, and I usually get a boo, but it's literally like be a moth Motel 6, right? Just leave a light on for them. That's really what it's about. But the key is not just any light. So if you flip on just a regular incandescent bulb, that's not going to work. You need to have lights that emit UVA light. So UVA light is basically like somewhere between like 315 to 400 nanometers. Right, is kind of the sweet spot. 365 is like really the key. So we use these like compact fluorescent bulbs that just let out that 365 to 395 nanometer wavelength. You get a lot of moths. It's as simple as like this is just like a hamper you could buy at like Walmart, right, for $2, you know, the college kids have or whatever. You could hang it over your porch light and put some egg cartons in there for the moths to kind of, you know, do their thing and flip the light on at night and see what comes in. Or you know, you don't need these fancy ones either. You can kind of go get these party bulbs even from Ace, you know, and these work fine too. These have a coating on them that filter out the light, so maybe they're not as effective, but they work. Or you can set up a light sheet, right? So this is a trick of the trade, you know. And it's funny because we're still using like 19th century, 18th century techniques. I mean, there's nothing to it. Like, I mean, it's just literally you put a light on it. And as long as the wavelength's right, you're going to get moths. But you can go fancy if you want with a light sheet and a mercury vapor light, or build bucket traps. I brought one of our traps that we use over there in our surveys that I have built. It's literally a 5-gallon bucket with a clamp light fixture, some PVC, a funnel. You screw the light in, you turn it on, the moths go into the bucket, and that's about all there's to it. So going back to that ease of sampling, right? It's really easy to sample these things. And you can, I think there's a few more pictures. But, and these mercury vapor bulbs, which are hard to find, they let out like true UV light. So they're covering the whole spectrum. And so, and then click again. So this is a light sheet, right? So a lot of these are caddisflies, but these are a lot of them are moths too. And so a lot of times what we'll do is we'll set up one of these, and this is nice because you can observe the moths at the sheet. You can take pictures, you can do things like that. But that's kind of all it is, right? And so it's really as simple as just getting one of those lights, put it in your back porch, flipping the light on. There's a guy, Carl Barentine, he's a retired biology professor in Spokane. He's great. I suggest if you do Facebook, get on the mothing and moth watching page and look up Carl. He posts his daily quartet. So he's been mothing, like he moths every night in his backyard from March 1st till like November 1st. And literally he's got, just some, traps out and lights and stuff. And he's documented, I think, 900 moth species just in his backyard south of Spokane. And he photographs them, he doesn't collect them or anything. But that's a lot of like what people do is moth photography is a big thing, right? And so if you're gonna do that, you need just a macro camera. So if you already have a macro lens or something, I use these little Olympus Tough cameras, which they're great. They focus to 1 centimeter and it's point and shoot. It's got a little LED light you can put on it. They're really nice, but if you have a fancier setup like Christy.
Hey,
here,
go back to that one picture a second.
Oh yeah.
I want to take a picture.
Yeah, I mean they're not like super cheap. Carl uses it's like a Pentax. It's like an ancient old Pentax that he uses, and he gets great pictures. This is a photo I took with that camera, It didn't even have to zoom, just hold it up. And it's got built-in photo stacking and stuff. There's another picture I took with this. That's like maybe a 4mm moth, but I took a picture with that camera. But if you already have a macro setup, you can do this. The trick is moth scales are really reflective. So if you're doing this at night, you need some sort of like speed lights or soft LED light with kind of a filter kind of thing. Or honestly, these days you just use your smartphone. I mean, some of these new Apple smartphones, I mean, this is like I took with like my old SE phone with a clip-on magnifier thing, but nowadays you can just, and Carl, what he'll do is he takes the moths out of his trap in the morning and he puts them in ramekins, puts them in the refrigerator for like an hour to calm them down, and then he photographs them in the nice, soft, morning light. But it's really fun, right? It's really fun. So now this is where we make the link. I'm going to try to make this link between mothing and bird watching, right? So finding moths is it's really similar to like finding birds in the sense that certain moths occur in different habitats, right? So you can go to a marsh, you can go to the pine forest, you can go to an alpine meadow, and you're going to find different moths in those areas, right? And there's also we talk about like the gestalt, right? Or the general shape of impression of size and shape, right, of a bird. Well, it's the same thing with moths, right? And so it might seem overwhelming at first, but moths, for example, you've got this like kind of fuzzy, moth here with rectangular wings. That's noctuity, right? And then your geometry are more like butterfly-like. They're generally delicate little spindly bodies. Your tortricity are generally small, and so on. Your crambid moths, what they call the snout moths, have their palps extended versus curled. And so each family has its own general impression of shape and size, right? And so if you kind of like learn that, then you have a starting point, right? Not unlike with a bird, right? If you look at it like, okay, it's this big, it's that big, it's got this mark, whatever. And then if we dig even deeper, moths also have a topography to them, like birds do, right? So we've got all this stuff. We've got the forewing, the hindwing, the orbicular spots, the discal spots, the subterminal bands, and all this. And so it's really a similar process. Finding and identifying birds, albeit you sort of have to photograph a moth or have it in your hand versus like a bird. You can kind of like look at it through your binoculars and get that sense. But there's a lot of similarities between the two, and there's a lot of resources. And so the Moth Photographers Group out of Mississippi State is kind of our unofficial clearinghouse of moth records. And what's cool about this is, I think if you click one more, if you go to the home page, click Plate Series. And then you click on this thing, try walking through the moth families. It brings you to these plates where you can just click on these plates, and it's by family, right? And so then you can start learning that general impression of size and shape, right? And start seeing, so when you do see a moth, you're like, oh, that's a noctuid moth, that's a geometron. That gives you a starting point to then be able to identify it even more. Now, Moth Photographers Group, most of the plates look like this, which isn't super helpful. But they do have, if you click on these little numbers here, go ahead, it'll bring you to a page like this, right? Where they generally have like in situ pictures that you can kind of compare. And then you can like look at these range maps and things like that and say, oh yeah, that thing's probably found. Another great one is the Pacific Northwest moths, who Lars Crabo has done. This is a really informative website in terms of their species pages, like have really great descriptions of like, the orbicular spot is round with a central orcellus and this and that, and really a solid description of that. And it gives you some habitat information. It also has phenograms. So these are super helpful, right? So if you see something, in these months and you think it's this, you can look at this and like, probably not, right? So it's really helpful. But this is another good website to get started with, identifying moths. And they have a great glossary, right, that you can, at the bottom of their homepage, that kind of explain all this. Yeah, go ahead. And then Bug Guide's another one. So Carl Barentine, he puts all of his stuff on Bug Guide. I think, click again. Yeah, so if you just click the little icon there, it'll bring you to the moth pages, and then you can search from there. But just some resources to identify. Now, of course, I say all that, and you can just get one of these AI apps that do practice. So this is the LEPS by Field Guide. It was developed as part of the LEPNet project, which was funded through the NSF. There was a big thing about digitizing collections and stuff, and they developed this AI app. And this is actually, you know, it's pretty handy. So you can literally just take a picture with your cell phone, upload it into this app, and it'll spit back, you know, some of the most likely things. Here's just you know, what the page looks like. iNaturalist works really well too. You can link LEPS to iNaturalist. Go ahead. And then there are a couple others that, this one's kind of almost defunct now, but just some resources. So now I know I gotta speed it up, don't I? Yeah, so I'm gonna speed it up. So I just talk about the Montana Moth Project to finish this up. So this is our flagship program. We started this in 2020 to document the diversity and distribution of moths in Montana by sampling moths in each county in the state. And it all started with barcoding and bird poop. So I was working with Kate Stone at MPG Ranch. We were working with poor wills and nighthawks and things like that. And we at some point we were like, oh, we were tracking into their roosts, and we were discovering they were leaving behind these really nice little fecal pellets. And that's when we discovered this barcoding thing, and we could amplify the insect DNA out of their poop and things like that. Well, then we quickly learned that our results were only going to be as good as the reference libraries that we compared it to, right? And even though the Barcode of Life database out of Canada is the largest and has millions of sequences, it's still kind of lacking for insect barcodes. And so we said, well, why don't we build our own reference library on MPG Ranch? And so that was part of my job, was going out and collecting all sorts of insects, you know, moths, beetles, butterflies, whatever, you know, pulling legs, building that reference library. And in that first year, I think we barcoded like 300 and some species of moths on the ranch. We did it again in like 2018. We barcoded several hundred more. And my, whatever gears are in my head kind of started like turning, and I was like, oh, well, this is, you know, I sort of had a casual interest in moths because I studied fly-minded owls, and they eat a lot of moths, and it didn't dawn on me that the diversity was that great, right? And so I started like thinking about, well, what's going on with moth research in Montana? And bottom line is we published a little study out of this, you know, comparing these with this library and things. And so it was around 2019, I started posting some of my records to iNaturalist and things like that. And I call this the gateway moth, this Simpistus sokar, which is the first, at the time, it was the first voucher record of this moth in Montana. I got a phone call from a guy, Chuck Hart. He does a lot of stuff in New Mexico, and he always likes to. He gave me permission to use this. He always likes to stop and visit me. But so Chuck called up and he's like, "Hey, dude, can I use this record in my season summary? I'm the Rocky Mountain coordinator of the Lepidoptera Society. I'm a research associate at C.P. Gillette Museum in Colorado State." Oh, yeah. And he's like, oh, and by the way, you know, there's nothing known about moths much, yeah? Like, it's like a black hole. I'm like, well, guess what I got going on, you know? So we talked more, and we said, okay, in 2020, this was COVID. We were like, okay, well, I've got this monitoring program, these yellow icons at MPG Ranch. How about we add, you know, these other sites here, and I'll set out one trap, and we'll do it once a month, and I'll send you the specimens. It'll only be like 3,000 or 4,000 specimens. I'll buy you a cabinet and drawers and stuff. And, well, at the same time, my tech that was helping me, was asking, well, can I take a trap to Red Rocks Lakes? You know, because she's camping all the time. Can I go take it? Oh yeah, sure, yeah. And I was kind of doing the same thing. Pretty soon we were turning up like first voucher records for the state, and we started traveling around. And here's Chuck. So at the end of that season, I don't know if that's a look of extreme concentration or exasperation. You know, this is COVID, so he's the only one in the. But we had sent him something like 8,000 to 10,000 miles by the end of that summer. And so we got together at the end of that summer and pretty much decided that we're going to have to do this in every county in Montana, right? And that's kind of the official birth of the Montana Moth Project. And then Chris Grinter from Cal Academy got involved, and then Marion Kirst, an entomologist out of Billings. She just recently moved on this year, but she helped out building the program in that area over the last few years. And so in 2020, it kind of started like this. We got sample locations. And so the year 1 through 5 was we were going to get moths from at least one location in each county. And then now we're in the second phase. And then it kind of moved in, you know, in 2021, we had these counties, 2022, we had a few more counties, and so on into 23, 24. Now this needs to be updated. We've added a lot more dots, but all these red dots are now our sampling locations. In 2024, we achieved our goal of at least one sample location in each county. As you can see, Montana is a huge state. We have a lot of work to do. If I were to, we have a lot more dots from last year to put up here. But now we're in that second phase. Now we're going back to each county to get a different location. And we want to sample like the kind of the predominant habitat in each county. But we also have been focused on targeting, oh, sorry. This is where we're currently at. Click it one more time. So we've got this is more like 60,000 now specimens down at the museum that are all database or vouchered and databased. Approximately 1,300 macro moths, so the big moths. So there's this division between, we won't get into it much, but the small ones and the big ones, right? We've got about 1,300 macro moths. The micro moths, there's a general rule of thumb that if you have, and our sampling curve is getting up there with the macro moths, like we're starting to top out. We still probably have a few hundred left, so the rule of thumb is there's probably 2 to 3 times at least many micro moths as there are macro moths. So what we anticipate by the time we get done with all this and identifying stuff, that Montana is going to be home to, I mean, I don't think 3,500 species in Montana is pretty reasonable. You know, when it's all said and done, so you know, an immense amount of moths in the state. And so we tried to, you know, sample the common habitat, but we also tried to, you know, focus on like unique habitats. So this is the Pryor Mountain foothills, super unique. If you ever get a chance to go to the Pryor Mountains and the foothills, it's our only cold desert habitat in Montana. You know, it's where the wild horses are around there, and then there's scorpions and sulfugids and camel crickets, and just really super cool stuff out there. And we find some unique moths out there. The Centennial Valley Sandhills, this is a Nature Conservancy property. It's one of only 2 sites. This one in the Centennial Valley, and then the Medicine Lake Sandhills that are only the 2 vegetatively stabilized sand dunes. So these are literally just sand dunes with vegetation on them. Like if you were to dig a hole, this is just a sand dune. Really unique moths associated with that. And in fact, this Copelepharon is a dune obligate moth that we found in droves down there. The Pine Butte Fen, another conservancy property outside of Shoto. We found this Hippocena, that is Striga, I think it is. It's sort of an obligate to this fen type of habitat, the first voucher record for Montana. Bitter Creek Badlands outside of Glasgow. I had no idea they even existed, but it's the largest. It goes from just north of Glasgow all the way into Canada. This Rhiza gratis. We find, you know, this unique moth out there, which is another first record. Closer to home, the Ross Creek Cedars. we've done that, and the Simpistus youngi, which is kind of associated with these really mesic forests. The acid shale forests in central Montana, which are ponderosa pine forests on this acid shale. We find this unique Uzoa oberfolii, which is another first voucher record. And so I'll just run you through a few things here. So we've been able to really kind of go ahead. This was the current map. This was one we found down in the Prior Mountains, this Emergestus consimilis, so you can see here where we've Shinia snowi, kind of the same thing. We've kind of brought that range up here. Now, I hesitate to call any of these range expansions because I feel like expansion implies that we knew what the range was to begin with, which I think, if anything, we're showing in this study or in this moth project is that we really don't. But this is a really rare find, one of maybe only a few specimens known that we found in the Prior Mountains too, another kind of southwestern sort of thing. Here's sort of maybe more of an eastern sort of thing that we found, and we've got a few more locations for this now. We talked about this copolepharon that we found in the Sandhills, kind of a west coast thing. This feeds on stinging nettles, I believe. But we found that, let's click it one more time, we found that in the Highwood Mountains, I think. Should pop up. Yeah, right there. So we've kind of filled that in a little bit there. And then, things like this biocontrol moth that was actually introduced into Montana in the 70s, but nobody ever tracks these things, right? And so before we added all these dots to the map, there was literally Bob Martin's dot near Helena, and that was it. And so we've been able to fill in all these dots with just a kind of a common moth. And then, of course, the micro moths, which I talked about, and you'll see some over there. I really enjoy spreading these. These are difficult to identify. A lot of them need dissections and things. But we've turned up some pretty unique ones. And I'm probably going to slip my mind what some of these are. Oh, this is a Eucosma crasana. Go ahead. And just, so we're doing all the moths, right? Keep going. Yep. So just the micro moths. This is one that Marion caught. And so all these get sent down to CP Gillette Museum, and this is Chuck. The process is they get sent down. We have students and things. They all get spread, and they get labeled, and then they go into drawers, unsorted drawers, and then we sort them into morphospecies, and then we start putting names on them, right? And once we get names on them, here's some curated drawers. Then those go to the databasers. And so all this information now is, this is just a look at some of the team that's been down there. We've been able to engage. So all this gets databased in this thing called ecdysis.org. So all this is publicly available. So you can log on to this and look up, moths in Montana or whatever, and you'll see all our records. We're working with the Montana Heritage Program. Currently, we've shared about 25,000 records. They're now going through the process of getting conservation ranks on some of these moths, doing range maps, creating habitat suitability models where we have enough data, things like that. None of the natural history information or pictures are really on there yet, but we're working. The idea is someday you'll be able to log into the Heritage Program and have all that information. Okay, go ahead. And so just to wrap it up, excitingly, we've discovered a couple new science species out of this whole effort. And so this was one that Marion collected at the Petroglyph Canyon outside of the Prior Mountains there. She collected this in early May. Let's see if we can back it up. In early May, and we were all kind of stumped by it. We sent it to Lars Cravo. He dissected it. Sure enough, it's new. Protogigia, and Marion Lars was gracious enough to let her name it. And so this is Protogigia priorensis, so it means from the priors. And there's a new species that got published a couple years ago that it's only known, well, it's since been found in the dune areas in Nebraska, but Montana's sort of its birthplace, if you know. And then a few years ago, in April, I was out in the Pryor Mountains and I found this moth, and I knew it was a Chesiodontis, but it didn't make sense because all these other Chesiodontis were from like California and stuff, and we didn't know, so we sent it to Cliff Ferris. He dissected it, and I don't know why he didn't let me name it, but he did. But anyway, this is Chesiodontis montanaensis, so from Montana, so a new geometric moth. And these are big moths, right? So these aren't like, you might be able to find a new moth in your backyard. That's what's cool about moths and insects in general. And then we also have several others. There's another one I found outside of Roundup on some BLM land that I just have the one specimen for, and Cliff dissected it. It is new. I've been back over and over and over. It's kind of an enigma. I can't find another specimen. But then we also have several more that are presumably new. We have one that turns out we find it all over the place. It's an early spring moth. It's new. We're working on describing it. And so not only are we expanding the known distribution and diversity in Montana, but we're able to find some new things. And I'll wrap this up. And so now we're just going back to all the other counties, adding second locations, and preferably in a different season and month. There's a lot of day flying moths we still have to find. I mentioned that. One thing I'd like to mention, and I'll have a slide with my contact information, but we're in this phase where we're really trying to find people that would be willing to have a trap, and you know, if you're going out camping or if you're in there, would be willing to kind of help us out with some of this. And so if anybody's interested in that, please reach out. And then we also have big ambitions, right? Eventually we're going to publish a big checklist for the moths of Montana. We're going to do some taxa-specific stuff and continue to do, you know, the pollination work. But here's just some of the fun that, you know, the volunteers and things have had over the years. And with that, sorry if I kept you very, hopefully it's not too late.
That's great.
Yeah. Yeah, just a few people to thank, and then go ahead, and here's the references, we don't have to worry about that. Click again. That's Christy Du Bois. Does anybody know Christy? she's a big moth, right? I took a picture of her taking a picture. And then here's my information if anybody wants to reach out. But yeah, with that, thank you so much for letting me talk about moths. And yeah, thank you. If anybody has any questions, I'll be happy to try to answer them. Yeah.
So, the pigment of the moth or a butterfly, is that a pigment or is it some kind of hair that does the light?
So the question is the color come from pigment or is it more of a structural thing? It's structural. So it's all about the way the light refracts off of the scales. Yeah. You know, the insectarium in Missoula just had a cool thing with one of their butterflies where they just changed the angle of the camera from like here to here, and it went from like blue to almost black, you know. And so it's yeah, all structural with the way the light refracts is really the color. Yeah. Yes.
What comes from the generation that's lost or bad because they little sweater.
Right.
This is a reason to see this in your presentation because I guess further work.
Yeah. Yeah, moths are good. By and large, they're good. They can be annoying. You know, the ones that eat your clothes are annoying. There's no doubt about it. But by and large, they're way more good than they cause problems. Yeah. Yeah.
Most of them overwinter as chrysalises, or you said some adults overwinter.
Some adults, but yeah, most of them will overwinter in their pupa. Yeah, and a lot of times most like butterflies do it kind of like they'll hang themselves, you know, from vegetation. A lot of moths they'll go into the soil and that's where the pupa will hang out.
And that's what the robin is looking for when it comes to
Could be, yeah, it could be, yeah, for sure. And there's even a lot of caterpillars that you'll find in the soil and stuff too. Like especially a lot of these cutworms I was talking about, during the day, they'll kind of go into that first layer of soil, and then they'll come out at night and feed, and then they'll go back into the soil during the day. But yeah, most of them do it as pupa. Yes. Yeah. That's interesting. I'm not sure. I can tell you that their antenna is, the males is where their chemoreceptors are. That's how they'll actually smell the pheromones. But I'm not sure about the club. That's a great question. Yeah, I'm not sure about that. You stumped me on that one. Yes. What percentage are wingless? It's a pretty small percentage. Yeah, I don't know if I could put a. I mean, if I had to guess, I would say less than probably 2%. Are wingless. It's not very many of them. Yeah, so the females are just on the hose plant calling, and the males come in, and then it's interesting in that regard of like, you wonder about like how do they get like genetic flow, right? If the females are like always on the same plant and area, I suppose a lot of different males coming. Yeah, but it's not a very big percentage that are wingless, just a few. It's more of the exception. Yeah. Yes.
Is there an average lifespan, if that's even possible?
Is there an average lifespan? That's a great question, and I get that question a lot. Talking to like Dave Wagner and some of these experts, like probably most people tend to think that probably about a couple weeks, for most moths. But versus like, you have like the army cutworms, right? Which they're flying now, they go up into the scree fields all summer and they fly back in the fall, and so they're alive for like 6-7 months. But on average, it's probably, according to the experts, it's probably 2 or 3 weeks, for the average moth. Yeah. Yes, Steve. That's a great question. A lot of the museums are struggling. It just depends on the space. A lot of these things they'll be if you put them in the right drawers and things, they'll sit there forever, right? It's just the challenge is are they ever going to get worked, and curated? And there's a lot of museums that just have like drawers and drawers of bugs that don't have any IDs on them, things like that, and it's a real challenge. And we've struggled with that too. I mean, we've operated on a shoestring with this, But fortunately, CP Gillette is a very active museum. A lot of students are coming in. We're able to get a lot of volunteers that are, that help us process that on the front end versus just throwing them all in the drawer and we'll get to it someday. But it's a huge challenge. The few years ago when they had the LEP grant and things like that, there was a huge push to digitize a lot of the stuff, and there's a program now called iDig Bio or iDig Bees, and so a lot of the museums are digitizing their bees. So I would say, if anything, there's been a shift to try to digitize stuff versus actually working the physical material. Yeah, well, there's a lot of private collections out there too. A lot of private. Steve Kohler. I don't know if anybody knows Steve Kohler. He worked for the Forest Service. He was a butterfly expert. He did something similar with butterflies in Montana like years ago, and He's got a private collection that's, I think it's 1,200 drawers. You know, and of like, you know, entomology drawers, they're about, you know, they're about this big, you can fit maybe 300 specimens in a drawer. He's got 1,200 drawers. And they're, most of these private people, they'll send them to an institution eventually. He was gonna send it to Florida, the McGuire Center, but I think we're trying to convince him to send it to CP Gillette, because then we would have probably the most comprehensive Lepidoptera of Montana collection in one location ever. But there's a lot of private stuff, but mainly institutional. Yeah.
So I have one last question.
Yes.
Can people visit the MPG Ranch and see some of these collections?
Can they visit? No, but I am working on, so we're working on bringing a synoptic collection of the moths home to Montana, and hopefully it'll either be at the Montana Natural History
Oh,
Center
nice.
or the Insectarium in Missoula. And so we're working on that.
Well, keep in touch with us.
Yes, yes,
And let us
that-
know when that happens,
You
because
bet.
we all want to go down there, don't we?
You bet, yes.
So if you have more questions for Matt, stay after and talk with him. Other than that, thank you so much. Let's give him a round of applause.